Dr. Verma for providing me this opportunity to chair this second technical session of the symposium being organized here in this Department of Zoology. Today, this session begins with the talk by an eminent geneticist, teacher of many of the teachers. He is Dr. S.P.Sinha-a. He served this department in the capacity of right from lecturer to professor to head of the department, the dean, faculty of science, this and that. But to us, he is sir. And to acquaint with the budding scientists, my students, be careful in listening his talk. Though it is written as the topic genetic basis of biodiversity. Biodiversity encompasses three different terms, three different aspects: the ecological, the taxonomic, and the genetic biodiversity. And he will enlighten us the genetic basis of biodiversity. Without going much into the detail about his work, I would like to invite my teacher, not mine, but to.
And it is about 14 or 15 years that I retired from the service. I lived, and now I am living at Patna, and therefore for the last 15 years I have not done any type of experiment or guided any type of research or wrote any research paper. So what I am just going to tell you is the summary of findings of my research scholars who are now faculty members of different places, and just at the end of that I would like to say that how the biodiversity can be explained by the help of that. May I have the dear students, if this is the original population, you know that there is a Hardy-Weinberg law which says that in a large Mendelian population, the frequency of alleles and the frequency of genotypes remains constant generation after generation. Now this constancy of frequency is just opposite of evolution, which says that evolution is a gradual change. So the constancy is also proved mathematically, and evolution is also proved every day. So how the two opposite forces are acting? Hardy-Weinberg law genetic equilibrium says that there shall be no change in the allele frequency or in the genotype frequency, and evolution says that there is always a change. So the completely two opposite forces are there operating, and both are true, completely true. How does this happen? This happens very simply that in every generation a new equilibrium is established, and there are certain factors which disturb this equilibrium, and that disturbance in the pre-existing equilibrium is one step towards evolution. And in the new generation again a new equilibrium is established, that is again disturbed, and again one step towards evolution takes place. So both factors are there. What are the factors? that are actually disturbing the equilibrium in each generation. Now, this selection, this is the rate of selection, due to the frequency of the recessive allele, you may not go. Selection means there are in the population a large number of individuals are there, and among those individuals all are not equally fit. So depending upon the environment, by the process of selection, unfit individuals are eliminated and the fit individuals retained. A question was asked to one of the colleagues of Charles Darwin that who is fit? And the answer was the one who survives is fit. Survival of the fittest, the meaning is that the one who survives is fit, not who is physically fit, monetarily strong. All those points are not there. So selection is there. Now there are mutations, and a large number of mutagens are there in the environment, both spontaneous as well as induced ones. Yes. Next please. So what we did, that in our laboratory for the last 20-25 years, we used different types of pesticides which belong to the organophosphorus, organochlorinus, carbamate. All these different types of pesticides were there, and these pesticides induced different types of mutations and chromosomal breaks, disturbance in crossing over, etc., chiasma formation, etc. And just I would like to take the name of some of my research scholars who actually did their PhD on this thing. First one is Dr. Bansi Prasad Bhagat, who retired as professor and head from the Vikram Singh University, Bir Kumar Singh University. Then there is one Alak Kumar Sinha, who retired as principal of SKR College, Bardigha. Then Amrendra Jha, unfortunately he is now no more. He was at Sabor College. Rajni Ranjan Prasad Sinha, who is in Magad University service. Govind Mohan Mishra, who was at the Sultan Medical College. Ashok Kumar Jha, Ashok Kumar Jha. One thing which Ashok ji said is very interesting, that these pesticides induced damage. They disturbed the chromosomes, damages were there. They induced mutations, as we tested Drosophila melanogaster. But Ashok ji, one thing very important you could find, that there is no difference in the frequency of the rate of crossing over. Pesticides were inducing chromosomal damages and mutation, but they were not increasing or decreasing the rate, rather decreasing, we may say, they were not increasing the rate of crossing over, and at the same time we could find that there was no difference in the frequency of chiasma formation in mice and grasshoppers. We were very much, under tension that when mutation rate increases, chromosomal breakage increases, but why the crossing over rate is not increasing? Why the chiasma frequency is not increasing? We were just thinking. Then Ashok ji said, one thing, sir, a very simple explanation is there, that crossing over, chiasma formation, etc. are the symbol of combinational variations which are very important, and by suppressing the rate of crossing over, the combinational variation is being decreased. So on one hand it is decreasing the variation, and on the other hand it is causing damage to the genetic material. This work was very much appreciated and we got it published in international journals. And there was some doubt in my mind that whether everything is there correct or not. So I repeated this experiment with the Ramesh Kumar Singh, who perhaps left this place, and he also found exactly the same thing. Of course, the organisms were different. The rate of crossing over of some other types of genes, etc. were there. Near the centromeric region or in the telomeric region, all those things were there. And one thing I would like to tell, Ram Pradesh Chaudhary, he also did a lot of work on different pesticides. So on these pesticides, we studied the effect. Now when these effects were studied, one logic came to our mind that if pesticides are causing damage to our genetic material, decreasing the recombinational variation by not affecting the rate of crossing over, but at the same time pesticides are very essential for our agricultural purposes. So we started a work that let us find out a dose of pesticide which is very small, and at the same time it is able to kill the pests. So it may be effective as a pesticide, and it may not be injurious to our genetic system. This work was, so I should say, this was searching for the threshold dose of the pesticide. The pesticidal property remains, but the cytotoxicity, the genotoxicity property no more remains. This work was done by Dr. Dinesh Kumar, who is presently Professor of Life Sciences at the National Council of Educational Research and Training, NCRT, Delhi. And on deputation, presently he is Joint Commissioner Academics of the Kendriya Vidyalaya Sangathan. He is doing work very nicely. So these persons were doing work. Now, after these pesticides, we came to some other types of additives, say for example the fertilizers. Dr. Om Prakash Chaurasia. He studied the effects of about 5 or 6 different fertilizers, and all photographs etc. were there, and the chromosomal preparations, all of them were published in 4 or 5 papers of the Cytologia Japan. So all those papers were there, and so fertilizers are also harmful to that. Then we searched other types of contaminants. Aftab Ahmad, who is at Marwari College, he studied the effects, genotoxic effects of medicines which the diabetic persons take. And you could find that these chemicals also induce damage. Then what I should say, just पहले वाले से. Now genetic drift, mutation we could show genetic drift. That is very small sized population. It migrates. Very small population. It migrates from original place, settles at certain particular place, and at that particular place, all the, I should say, the marriage takes place among themselves. Some sort of inbreeding is there, and what may be the effect of this inbreeding? Dr. Muhammad Afzal, who is presently at Aligarh University, he has studied this. His PhD thesis was on the effects of inbreeding, which causes not only the mortality or mortality rate or decreased fertility of the females, but even he studied, should I say we studied? We studied the behavioral pattern, that is IQ test, intelligence quotient tests, both performance type as well as mathematical type. Those tests were there, and the children born out of this inbreeding, they had lower IQ. And the onset of certain properties, for example, after the birth of the child, when the child for the first time hears something, sound, sees a visible fixation, he goes on looking at anything, visual fixation, social behavior, all these things, on which date these things start, and it was found that the populations which were inbreeding, they were lower intelligence, delayed maturation of the inborn child, increased mortality rate among the children, and decreased fertility among women. So these are the properties which were observed in the human population. So the process of genetic drift, a small genetic drift was also observed in the population of fishes. Naguma Ahad, you remember this name?
Yes.
Professor Ahad was a principal of TNB College, Bhagalpur, and his daughter, Naguma Ahad, who is presently at Patna, in a particular pond. That pond, where that masjid is there? Shahjangi, yeah, Shahjangi Pokhar. From there, she time to time collected fish population, Clarius batracus, Ophiocephalus punctatus, etc., and she studied the frequency of the ABO blood group. Because in the fish also the frequency of the ABO blood, antigen A, antigen B, they also act upon, and it was found that all the fishes were either of the B blood group or O blood group. There was complete absence of the A blood group and complete absence of the AB blood group. It means by the process of genetic drift, only one type of alleles exist and no other type of alleles exist. Such is the phenomena. And then by the process of migration, in this process of migration, a large number of my young colleagues, they worked on this. Nasir Ahmad Ansari, who was at Sahibganj College, and perhaps he is at somewhere at the university headquarters. Nasir Ahmad Ansari, he studied in the Pahadiya tribes.
Already retired, sir.
Oh. Kamal Kishore Sinha from RD and DJ College, Munger.
Yes.
And one very interesting point of Kamal Kishore Sinha was that we took a population, don't mind if I am using this word, the Kayasthas of Bihar. And we calculated the frequency of the different genes, alleles. For example, the morphological characters, free or attached earlobe. There are so many different types of testing ability, PTC testing, PTC non-testing ability, and it is in this manner there were so many transferase enzymes, APO blood group system. We calculated the frequency of these alleles in the human population, in those populations of the caste. They were the Srivastav caste, current caste south of Ganga, current caste north of Ganga. Then Ambast and Srivastav, Ambast, Karan and Asthana. We studied there and we could find that, and on the basis of that we calculated the genetic distance between these populations. And it was found by him that the genetic distance among all these four, Asthana, Karan, Ambas to Srivastav, the genetic distance was so much there that they cannot come from a common stock. Our logic was that this caste system of castes has emerged out of the process of fusion. Persons belonging to completely different groups, when they started similar type of profession, they all were called castes. So in sociology also it is said that the castes originate by the process of fusion. Persons of different origin performing similar type of profession, they come and belong to the one group. And just opposite of this was performed by Kamal Kishore Sinha of the RDDJ College Munger. He studied among Vaishyas, Kalwar, Teli, some 5-6 communities were there, and all of them belong to the same group. Original place is from Haryana and Punjab, and from there these persons came here, and the different castes were there. Dhamuk, Teli, all those there, Kalwar, all those were there, and it was found that the genetic distance between these populations was very, very low. Means they were insignificant. Means all these different castes are nothing, but simply they belong to one and the same pool, they belong to one and the same population, just opposite of the caste. And then the other principle is there that what they happened, that when they came from their original place, someone started selling oil, someone started halwai, someone started doing a wine business, someone started doing the shop, rice, wheat, etc. selling, and by that the same population broke into different castes and the different names were there. So the origin of castes by fusion is proved in the case of castes, and by fission it is done in the case of the Vaishyas. That you could find, and both these two papers were published in the British Journal of Evolution and Journal of Indian Medical Research, Population Genetics. These papers were established. The migration. Madan Mohan Lal was the first person to study this population, and it was financed by the University Grants Commission. A large number of the populations of the Marwadis have now settled at Bhagalpur. And this Marwadi population is performing job here, and by the process of that migration, the physical difference with the local population was not at all there, but some genetic features were quite different from the local population. So, by the combinational variation due to random assortment of non-linked genes crossing over among the linked genes, by the process of selection, by the process of mutation, genetic drift and migration, the original population, which had a fixed frequency of alleles and phenotypes, gets changed. And these individuals, these of his progeny populations, they come and live in their different ecological regions, and the result is more efficient or neutral or non-adaptive interaction. It may be more efficient, it may be neutral, or it may be non-adaptive interaction with the environment. So, improved neutral or non-adaptive adaptations are there. And this very process is called microevolution. Evolutionary step at the very, very lower level. Microevolution. Next, please. I just already discussed this thing. So what is there? A founder population on the periphery of a species range is often subject to a strong and new selective forces. I just show that from the original population, when it has migrated, it no more remains that oval or circular shape, but rather so many different branching parts are there. So in that branching part, what happens? That either they undergo the pre-mating mechanism, isolating mechanisms, that is the habitat isolation, seasonal isolation, behavioral isolation, mechanical isolation, or the post-mating, that is there is gametic mortality, zygotic mortality, hybrid inviability, hybrid sterility, hybrid breakdown, and the result is origin of new species. These both pre-mating and post-mating mechanisms are the result of the genetic makeup of the population, and if a change is taking place in the genetic makeup, either by mutation etc. or by the isolation or by the migration or whatever factor may be, the result will be the origin of new species. And this origin of new species may be sympatric or allopathic. Next, please. Now this is the original area. There are two subpopulations, A and B, and due to microevolution of a species, what happens? That one population, suppose for example, comes here, and at that time there is reproductive isolation. And what may be the basis of that? There may be polyploidy, hybridization, apomixis, or sexual reproduction may change into asexual reproduction. Next slide, please. Just say, for example, this was done in the case of the cars, goose, which grows in the Tiara region, goose, Saccharomyces spontaneum, something like that name is there. What we find here, these are the chromosomes at the metaphase stage, and this is the nucleolus. The number of chromosomes, one pair, two pair, three pair, four pair, it is in this manner. There are 16 pairs of chromosomes in the primary spermatocytes. And at the later stage, what we could find, that the two primary spermatocytes are fusing with each other by the formation of a cytological canal or cytological bridge, And from this particular cell, out of 16 bivalents, 8 bivalents have migrated to this chromosome, and only 8 bivalents are remaining. 1, 2, 3, 4, 5, 6, 7, 8 bivalents there. So, as a result of which, this particular cell from the diploid condition has become the triploid condition, and in this triploid condition, this is the metaphase of that one, and from this it is the tetraploid condition. So it is a third one is a recipient cell during pre-metaphase with 2 nuclei and double the number of bivalents, and a syndiploid is metaphase 1. So these are the chromosomes. This part is most important because here the pollen mother cells are fusing with each other. Next please. And they are also same degree of polyploidy is there. Next please. And one effect of this polyploidy is there, that in the control form, both female part of the flower, gynoecium of the flower, and the male part, the androecium of the flower, both of them emerge together, but due to the fusion of the pollen mother cells, when polyploid forms were produced, in them only the male parts are there and the female parts are completely absent. As a result of it, in the normal control form, there was a sexual reproduction and the population was growing, but here the male parts are different, male flowers are different, and the female flowers are different. So there is, in place of sexual crossing, there is intersexual crossing. Male plants are of one type, female plants of one type. So please back. So that reproductive isolation is there. Actually, the bisexual plants were having normal sexuality, but here the reproductive isolation is there because the male flower is separate and the female flower is separate. And this is caused by the process of polyploidy, or this is by the process of hybridization or apomixis. And so gradually, though they are living at the same place, sympatry, that is they are living at the same place, sympatric, and in that sympatric population, by these very processes, the two groups of the populations are formed, and this develops into one species in due course of time, this develops into another species in course of time, and therefore that is the sympatric speciation. So this is one basis how evolution may take place. And another one is that there is a geographical barrier between the two, which may be a mountain, say for example Himalaya, which may be a desert between two grasslands, which may be a big ocean between the two continents. A geographical barrier is there. So I should say it is ecological barrier. Completely two different ecosystems are there. Say for example the grassland ecosystem and the desert ecosystem is there, and one set of these, the organisms which are more fit at this place may not be fit at this place. Here the xerophytic adaptations are essential, and here the hydrophytic adaptations are essential. So original population was the same, but when they are living in two different ecological conditions, the population gradually increasing, and they are called as the allopatric. Allo means different, patric is the patric or whatever you may call it. So the allopatric, this one was the sympatric in the same population, and this one the allopatric. So even after some time, when these two populations come together and the barrier is removed, now they are genetically so different from each other that now it is not possible for any type of breeding between them. So even if the barrier is removed, it is not there. We have found it in the case of human populations also, that the Kalwar and the Teli and all these things and Halwai, all these are there, but that type of inbreeding is not taking place in them because they migrated, they separated at different times, living in different localities, and all these things happened. So our studies would also suggest this very thing, so that appreciation. Next please. At another level, fragmentation of pre-existing species involves the origin and evolution within a short type of great many adaptive types. Great many adaptive types. And this is called macroevolution or adaptive radiation. Adaptive radiation we know very well. So this type of adaptive radiation, well, as a geneticist, I am not able to study this particular phenomena because the ecologists and the geographers, they can very well say that why fragment, or paleontologists can say that why this different process. Same stock is there, but when they are living within a short time, great many adaptive types are there. So this is a fragmentation is called, and this fragmentation due to subdivision of the species into many groups, invasion of numerous new environmental situations, diversification of the structure and biology, and during this process may show evolutionary divergence, even convergence in special adaptations with other distantly related groups differing in their matrix of general adaptation. So what is there? General adaptations, then comes the special adaptations, then rigidly specialized super specialists, and every ever-changing environment, they are unable to adopt, those are unable to adopt. Extinction is there. So those who are able to adopt, they survive, and those who are unable to adopt, they get extincted. So main point is this here, macroevolution or adaptive radiation. Next please. And then comes the mega evolution. On rare occasions, in a new environment, old organs are put to new use. I stand subject to correction, Professor, if I say that an air sac of a fish gets modified into lungs. I don't know I am correct or right or wrong, because when we studied dipnoid, we knew that some sort of this arrangement is there. Pentadactyl appendage, the fins of fishes, stegoderms, got differentiated as the appendages of the amphibians, and that was the pentadactyl appendages. So they are one and the same thing. So origin of new biological organization, if the fin which is used for swimming gets changed into the pentadactyl appendage, it means further movement on the soil. So this leads to the origin of new biological organization of the plant. And characteristics are there, divergent lines of ancestral stock. One of them crosses the ecological barrier, and this breakthrough and shift are always rapid. New zone ecologically accessible, devoid of competition, requirement of new general adaptations for invasion, and the entrance into new zone and the mega evolutionary changes take place. Ambulata among the mammals, very common. Artiodactyla and Perissodactyla. मतलब आप कभी कंपेयर कर लो एक घोड़े का खुर और एक गाय का खुर. घोड़े के खुर में क्या है? बिल्कुल पूरा का पूरा ऐसे गोल है ना, पेरिसोडैक्टाइल. और आर्टियोडैक्टाइला 2 ग्रुप बना हुआ है, बीच में खुर फटा हुआ है, जो कि कैटल में, गाय में पाया जाता है। क्यों गाय बकरी क्यों? अरे घोड़ा का जो वेराइटी, दोनों मोर और लेस दे बिलोंग टू द सेम ग्रुप, बट those groups which were living in the dry land, for their movement and running, they had to complete this thing, because the divided one will decrease their force, and those animals which are living in, I would say, wetland or something like that, where the soil is more loose, the goat and the cattle, etc., there you can very well see that when they move, the mud which is there, which is replaced, comes out of this gap, and their movement is taking place. So population is the same, but the adaptation is different. So entrance into new zone, as a result of which the mega evolutionary changes take place. Next please. So the original stock is there, and nature is performing evolutionary experiments, and these experiments are basically of the genetic nature, either by the rate of mutation, or by the rate of crossing over, or by the rate of polyploidy or aneuploidy and isolation, etc. So the original stock, the nature is making a large number of evolutionary experiments. All of them are not successful. If suppose, for example, this succeeds and horse is becoming more fit like horse, Eohippus, Myohippus, Pliohippus, Plesippus, Meryhippus, so all these things are there right from the horses which existed in the Eocene time. They were called Eohippus. At the Miocene time, they were called Myohippus. At Pleistocene, they were Pleistohippus. So at different times, different types of horses were there, and gradually perfection of the horse was taking place. And finally, this is the evolutionary trend, a gradual perfection is taking place. And maybe that fails to adaptation and it becomes extinct. So either those who adopt, they survive and evolve. Those who fail to adopt, they are eliminated. Next please. This is simply the elephant's body size or the tooth or the foot mechanism at the different periods of time, Eocene, Oligocene, Miocene, Pliocene, Pleistocene, recent. So different times are there. The body size is this one. The number of tooth, low grazers is there. The 3 to 4 tooth there, and there the number is increasing. Next please. So what happens that evolution takes place? Microevolution is a sequential process. Adaptive, neutral and non-adaptive changes in gene combinations. Speciation is divergent adaptive changes in isolated related populations. And then macroevolution is of divergent nature, adaptive diversification and radiation, and mega-evolution divergent origin of major new adaptive biological organization. So these are the sequential changes, and divergence in the living world are the major sources of biodiversity. And both these sequential changes are due to the rate of mutation, crossing over, etc., etc. And the divergence is due to the isolation and the changes in the genetic makeup, etc. And that is the major source of biodiversity. So the forces and process of evolution lead to higher and higher levels of adaptation. And adaptation is the genetic basis between organisms and their existing environment. Next. So, as conclusion, I am of opinion to say that the nature is both producer by chromosomal changes, crossing over changes, by polyploidy, by intermixing, crossbreeding, interbreeding, whatever may be, the nature is producer and at the same time the nature is selector, and therefore the genetic basis of the biodiversity is there. So the nature is both producer and selector of biodiversity. Over. So this is the information that I shared with you all. It is you all people who did it. For example, say Dr. Parimal Khan, who is here, Madhulika Sahai, Dharmshila. They worked on one thing very special. Mycotoxins. Aspergillus flavus is a fungi, and this fungi infects our crop plants, and these crop plants we are eating. So what happens that many of the fungi produce different types of harmful substances, chemicals. Say for example, very common Aspergillus flavus, a fungi, when it infects our food, it produces a toxic material which is called aflatoxin, A for the Aspergillus, Fla for the flavus, Aspergillus flavus, and the toxin is there. So the Aspergillus flavus toxin is abbreviated as aflatoxin. So Madhulika Sahai did this work on the effects of the aflatoxin, and she could find that not only the chromosomal damages, but the cancerous growth, malignant growth in the liver, on the skin, the connective tissue, all these things happen. All these things happen
by aflatoxin.
By aflatoxin among the mice. Then Dharmshila, she took another type of aflatoxin, mycotoxin, ochratoxin. They also produce these types of changes. And they also studied how these changes can be minimized. Many works were done on the antioxidant vitamins like vitamin C or vitamin A. So all these works were done, and they could find that some sort of damage can be decreased. Damage can be decreased, no doubt, but the damage cannot be negated. Damage is always going to take place. So, the information which I shared with you, what is the value of this information? The information is this, that please beware or avoid as much as possible the environmental pollution. Because if you are not going to remove the environmental pollution, you are going to suffer from many hardships or difficulties from your genetic makeup, and as a result of which you see what happens, that you will be unable to adapt to the environment and the extinction will be your fate. So many different types of diseases are very often reported, whether it is due to arsenic or due to this or that. So many different chemicals are there. And one thing, it is changing our behavioral pattern also. Dr. Afzal studied this behavioral pattern. It is affecting our behavioral pattern also. And suppose those individuals who are unfit due to the genetic load in them, genetic damages in their genotype, for their survival. If they become criminal and start snatching and doing all these types of things, what will happen? Those who are the normal persons, their life will be in danger. So today, so much criminal activity that is going on, not only the different types of diseases or the cancer, etc., but I am of opinion that so much of criminal behavior is going on, which is making our life very miserable and very dangerous, may be due to the environmental pollution, because that is affecting our genetic material, and our behavior is also controlled by our genetic makeup. And those normally they should have been eliminated, but thanks to the surgery and the medicine and the vaccine, etc., they are being preserved, but their mental thinking remains the same. So if we are thinking to survive and prosper, it is very, very essential that we must protect ourselves from the environmental pollution, not only ourselves, for the whole environment itself. So it is that value of the information in zoology. Please protect, otherwise this Homo sapiens sapiens is going to be extinct in 1,000 years.